Good morning, my dear students. And today in this class, we are going to learn about what is transcription. Okay, and before knowing the process, we should know the term what is transcription. What is transcription? Transcription is just copying the information from one another. Okay, so what actually happens? If you are copying some information from board to your notebook, that will definitely differ from person to person. Have you all accepted the same kind? What uh, what are the molecular mechanism which will happen inside our body means we need to grow. Okay, so our first we should know the central dogma of our life. So what is the central dogma? Central dogma which will helps the which is the major phenomena for molecular biology. Okay, what it actually means that is DNA codes for RNA and RNA codes for protein. Okay, in detail, what it actually means: DNA to RNA from mRNA to proteins. So this is the central dogma of life. Okay. So what is the transcription? So as I told you already, transcription means you're just copying from information from one to another. So here the DNA is here, from DNA to RNA the information will copied. Okay, so when in replication, I told you in previous video, DNA to DNA is a replication process and DNA to RNA is a transcription and RNA to protein is a translation. So the overall process is called the central dogma of molecular biology. Okay, so in this chapter you must clear the concept. Okay, see, DNA is the storage of total information. From that, mRNA is the information carrier. This is the information carrier which will pass the information to one another. Okay. So the proteins, active cell machinery. So here the active cells are present. Okay. So hope this concept is clear for all. So we'll go for the next one. So what is the transcription? Which is nothing but the synthesis of one RNA molecule using one of the two DNA strands as a template by the enzyme RNA polymerase. Why here RNA polymerase is present? Because we need to synthesize the RNA from the DNA. Okay, here our DNA, from DNA to RNA, that is our transcription. So the synthesizing polymerases are RNA polymerase. Okay, as we all know, our DNA is a double-stranded molecule. From the double-stranded DNA, we just going to synthesize a single-stranded RNA. So synthesis of one RNA molecule using one of the two DNA strands as a template by the enzyme RNA polymerase. Okay. So see the diagram. So this is a double-stranded DNA helical structure, and this yellow is the RNA polymerase, and from this end to this green region stands for the promoter region, and this pink region is the transcription starting site. Okay, and this is the see here one hanging strand is there, and this is the newly synthesized strand. So when the newly synthesized strand is this direction means your template strand is automatically that side. Okay, so in this concept hope you should clear this. So we'll discuss in the following slides. Okay. Oh, see. See, For prokaryotes, DNA to mRNA, mRNA to protein. So this is the overall central dogma of prokaryotic mechanism. When here for eukaryotes, this white diagram is represent these mechanisms are present inside the cellular nucleus, and this blue represent the cytosol, which is a cellular organelles present outside the nucleus. Okay, so in this place your translation takes place here, and here your transcription. Okay, inside the nucleus only your transcription mechanisms is present. So outside the nucleus your translation is happen. So this part will clear. Okay, so and one more thing I need to tell you one thing. So see the DNA strand here. Prokaryotes there is no what introns or exons, non-coding sequence. Here only coding sequence are present when compared to eukaryotes. Okay, but here for eukaryotes some introns, it is our non-coding sequence are present. So that machinery, in this machinery we need to elicit the non-coding sequence, and then that is called the processing. After processing, say slicing, after that only your newly synthesized mRNA will come out. So these two processes are happens in eukaryotes only, not in prokaryotes.